Good morning, everybody. Today we're going to start lecturing on replication of small DNA viruses, those viruses that their genome contain DNA and their size is relatively small. Before getting to that concept, let's have a little bit of a review on some concepts in cell biology. I am confident that all of these terminology are kind of review for you, but we will go through them regardless. We have a eukaryotic cell cycle. These are a collection of events that prepare the cell to divide, so one cell becomes two cells. We will talk about cell cycle a little bit more in detail and its regulator and its contribution to cancer development. Cellular transformation, the next definition, are those changes in morphological or shape, biochemical, or growth properties of a cell. Remember before I talked to you guys about if we have a cut or an abrasion in our hand, for instance, the cells on both sides of the damage will sense the damage, start speeding up the cell cycle until the gap or that juncture is being filled. However, there is in normal cells have a property called contact inhibition. When they hit each other, they stop growing. However, one of the growth properties of cancer cells that has been lost is so-called loss of contact inhibition, meaning that even though they hit each other, they realize that they're contacting each other, they still pile on top of each other and form a tumor. And that's why cellular transformation or cancer is a disease that has abnormal cell division. There's no control over it, and it's usually characterized by the presence of a mass or a tumor. Then we have tumor suppressors. These are genes and the gene products, or the genes will be translated into proteins. Those proteins are regulators of cell growth, meaning that these will, once they are activated, they prevent the growth of or conversion of a normal cell into a tumor cells. Usually in cancers, we have silencing or turning off of tumor suppressors, which allows the cancer to grow and divide. Let's move to slide number 3. Eukaryotic cell cycle. The cell cycle occurs during interphase, mitosis, or cell division. Let's see what it is. Eukaryotic cell cycle is a tightly regulated process. There are a series of proteins that regulate cell cycle. These are called cyclins, and a group of enzymes, kinases, which are capable of transferring high energy phosphate groups, are called cyclin dependent kinases. Cell cycle has 4 phases: G0, G1, when the cells are resting; S phase, when the cell is being getting ready to synthesize its own DNA and double the amount of DNA; G2 phase, and starts mitosis; and M phase is when mitosis is completed and we have 2 daughter cells. Each of these stages, G1, S, G2, M, the transition through these processes is tightly regulated by specific cyclins and cyclin-dependent kinases. For example, we have cyclin D for G1 phase, cyclin E at the transition of G1 to S phase, so on and so forth. The term telomerase refers to specific sequences that are found at the end of chromosomes. Telomerase is the enzyme that is used to replicate DNA at the telomere sequences. And as the cells divide, telomeres become shorter and shorter by each cell division, until we get to a point that there is no telomeres. The cells stop dividing. They either undergo something called senescence or they just die. And cancer cells have increased activity of this enzyme telomerase that will act on telomeres and confers a continuous cell division property to the cells. Going to slide number 4, there is a little bit of a description for those of us who may have forgotten about the events happening during cell cycle or cell division cycle, and some information about telomere. I would strongly encourage you guys to take a look at these definitions. Slide number 5. Remember I said that there are some genes, tumor suppressor genes, that they control unchecked proliferation of the cell. So one of these growth control genes is called RB. RB stands for retinoblastoma susceptibility gene, and RB will what happens is, inside the cells we have some proteins that regulate transcription, meaning that these proteins E2F and DP stimulate the transcription of those genes, including cyclins, cyclin-dependent kinases, that are required for passage through various stages of the cell cycle. RB gene will be translated into RB protein, or so-called RB gene product. RB protein is also called P105RB. P for protein, 105 is 105 kilodalton, the molecular weight of the protein. So RB protein binds to these two transcriptional regulators, E2F and DP. Once RB binds to it, it inhibits transcription of those genes required for cell cycle progression. Therefore, it's very logical to stipulate that if there are any mutations in RB, it will result in uncontrolled cell growth and tumor. The tumor usually happens in retina. And why tumor happens? Because RB protein will lose its function, so the cells will continuously grow and divide. Moving to slide number 6, another protein that regulates cell cycle is called P53. P53 gene is turned on in cells that are that escape repression of cell division. So I know that this statement is a little difficult to comprehend. Let's see what will happen. P53 is also called guardian of the genome. what will happen with P53 is that when P53 gene is being translated, we get a protein that is 53 kilodalton called P53. It's a transcriptional regulator. So once P53 gets activated, three things happen. Number one is that on the cells that are dividing out of control, it leads to the arrest of the cells at the G1 phase of the cell cycle. Oh, the second consequence of P53 translation is apoptosis or programmed cell death of the cell. And the third is contact inhibition. Logically, inactivation of P53 results in loss of cell division repression. In other words, if P53 is mutated or inactivated, the cells grow uncontrollably. The figure on the left side of this slide shows that P53, the different mechanisms by which viral gene products inactivate P53, resulting either in its degradation or loss of function. And towards the end of the figure, lower left corner of figure, we see that once P53, which usually comes in tetramers, 4 molecules, once it's activated, it either produces a cell cycle regulator called P21 that causes the arrest of the cell, or it causes the expression of a protein called BAX, which will lead to apoptosis of the cell. So BAX is a pro-apoptotic molecule. Once it's expressed, the cell will die. So next slide, number 7, there I've included some information about the retinoblastoma protein and P53 protein. Please take a look at it and try to understand these two, understand the name, the gene, and the function of these two gene products. Slide number 8 is P53 pathway. In normal cells, under normal circumstances, P53 is inactive by its physical association with a negative regulator called MDM2, which stands for mouse double minute number 2. So physical association of these two proteins, P53 and MDM, results in inactivation of P53. However, if there is damage to the DNA of the cell, or there are some abnormalities in normal cell cycle progression, or under hypoxia or low oxygen levels, P53 will dissociate from MDM, becomes activated, and exerts its effects via two major mechanisms. If, as we can see on the left side, in the left rectangle below P53, if the extent of the damage to the DNA is little, cells will undergo temporary cell cycle arrest. DNA repair mechanisms will kick in. They repair the damaged nucleotides, and cell cycle progression will resume. However, if the damage to the DNA is so extensive and there is no way for the cell to control that damage or repair it, the cells will undergo apoptosis. And both of these two processes, cell cycle arrest, which repairs the damage, cells will continue growing, or apoptosis, which is the elimination of cells with extensive DNA damage. Both of these two processes contribute to cellular and genome stability. Just think about it. You do not want to have a cell with damaged DNA, maybe a misplaced, maybe a missing nucleotide to go through replication, because that uh, abnormality will be propagated and transferred to the daughter cells. So if there's a little damage, we take care of it. It's like a flat tire. If the hole in the tire is too little, we'll patch it up. But if the damage is too much, in the case of apoptosis, we need a new tire. We just get rid of the old tire. Moving on to slide number 9, animal viruses that have DNA as their genomes. The size of animal DNA viruses is larger than RNA viruses. They range from 5 to 200 kilobase per. These viruses act very similar to host genome, and in some occasions they use host cell machinery for the replication of their genome and transcription of their messenger RNA and translation. Inherently, we have two major problems that a DNA virus encounters, because DNA virus depends on host cell DNA for the replication of its own DNA. So what are these two problems? The problems are, number one, the host cell must be in S phase, the synthesis phase, where it can actually double its DNA. And the second problem is that host cell DNA is linear. DNA synthesis cannot occur at ends of linear DNA molecules. This is a problem that we call it end problem, and it's because of use of RNA primer by host cell DNA polymerase. I'll go into a little bit more details about these two issues. But again, a virus that its genome is DNA enters host cell, it needs to use host cell machinery to replicate more of its own DNA, make RNA and translate RNA, so on and so forth, that we have discussed extensively before. Since host genome is DNA, viral genome is DNA, there are two problems that virus encounters because it depends on host cell for DNA replication. These two problems is that, number one, host cell needs to be in the S phase, and number two is that DNA synthesis cannot occur at the end of linear DNA molecules, called end problem. So let's take a little bit of a closer look and see what is going on with these two processes. Move on to slide number 10. So the first problem that virus encounters is that it depends on host cell being during its S phase of the cell cycle. How virus overcomes this so-called cell cycle S phase requirement problem. 3 ways. One is that some small DNA viruses only affect those cells that have entered the S phase, such as parvovirus. Parvovirus only affects cells that are in the S phase of their cell cycle. Other viruses, such as adenovirus, papillomavirus, polyomavirus, once they infect the host cell, induce the host cell, force the host cell to enter S phase, then they can hijack the machinery. And third solution is that some large viruses, such as herpes virus or pox virus, bring in their own enzymes. Therefore, they are not dependent on host cell enzyme. So let's recap. Viruses, DNA viruses, when they come in, they require the host cell to be at its S phase of cell cycle so they can hijack machinery. How can they overcome this? Three ways. One is that some small DNA viruses, such as parvovirus, only affect those cells that are during the S phase. When you think about it, not all of the host cells are synchronous, meaning that they are not synchronized to be all at the same phase of the cell cycle at any given time. Some cells are at G1, some cells are at S phase, some cells are at M phase, some are at G2. So there are different stages. Some of these DNA viruses, such as parvo, only affect those cells that are at their S phase. Some other cells, upon infection, induce or make the host cells to go to the S phase, and some viruses, larger viruses such as herpes or pox virus, really don't care about which phases of cell cycle the virus is. They encode their own enzymes, and therefore they do not need host cell enzymes for DNA replication. Moving to slide number 11. The second problem that DNA viruses face upon infection of a host cell is end problem. Remember, we mentioned that the DNA cannot be replicated at the end of linear molecules, linear DNA molecules, and the reason for that is the use of RNA primer by host cell DNA polymerase. So how can we overcome this? Very simple. One method is that instead of using RNA primers, we use protein primers. The second way is that to overcome that linear end of the DNA molecule, we use rolling circle model that has no ends. So circular genome forms concatenamers, which I'm sure you guys have been familiarized with the concepts before, but I have slides that walk you through the whole process for extra information and for your knowledge. The third mechanism is that some viruses resort to reverse transcription or RT, and they use the enzyme that is being used is host cell telomerase. In this instance, we make RNA molecule and then convert it back to DNA molecule. This we're talking about viral DNA. So three solutions that viruses take advantage of to overcome that end problem. The first solution is instead of using RNA primers, we use protein primers. The second solution is that, remember we said that end problem is that the inability of DNA to replicate at linear DNA molecules, which is usually towards the end. So in order to overcome that, we form concatenamers that have no ends, and we join these concatenamers together through the process of rolling circle model. The third solution is to resort to reverse transcription. Host cell telomerase will convert RNA into DNA molecule. Moving on to slide number 12. There is a description of rolling circle replication and the definition of a concatemer. I am sure that you guys have encountered these before during the prerequisites for this course, but for simplicity and availability, I posted this slide so it's readily available for you to look at. Moving on to slide number 13, again, rolling circle and its use in virology. Moving on to slide number 14. I guess we've gone over this a few times, and it was on your first midterm exam as well. Range or patterns of viral infection. There are 4 patterns: asymptomatic, where there are no signs and symptoms of the disease; acute, where the clinical signs and symptoms of the disease are apparent; persistent, which is long-term. Persistent is subdivided into chronic and latent. Chronic is a form of long-term viral infection where infectious virus is present, whereas during latent infection, we do not see infectious virus. So there is no viral replication going on. Virus remains dormant in some cells and becomes reactivated. The 4th pattern of viral infection is transformation. When the virus comes in, it will alter the normal dynamics of host cell. The result is the production of tumor, so it transforms the host cell. During transformation, we do not see an infectious virus, and what we have is the viral DNA, either part of it, partial DNA, or the whole viral DNA genome. Moving on to slide number 15, we have talked about the virus replication cycle. These steps include attachment and absorption of virus. Virus through specific receptors will bind to host cell, will get into the host cell, gains entry or penetration, and uncoating, which is separation, either partial or complete, of capsid from viral genome or nucleic acid. Then we have gene expression and protein synthesis. This step is divided into 3 stages: early gene expression, early proteins, usually non-structural proteins, enzymes required for the next step, which is genome replication, and late gene expression results in the production of mainly structural proteins. So here we have capsid proteins and a lot of genome. These two need, through specific interactions, assemble or mature. Afterwards, the virus is being released from the cell, either through lysis or through budding. Remember, we said that during budding, virus will pick up a small portion of host cell plasma membrane as its envelope. Also, budding may or may not lead to the lysis and death of the host cell, whereas lysis kills the host cell. Moving to slide number 16. The very first family of small DNA viruses is family Polyomaviridae. These are viruses of animals and humans, meaning that they can use both animals and humans as their hosts. Specific examples of viruses within this family are polyomavirus and vacuolating virus. They have circular double-stranded DNA genome, 5 kilobase per. The DNA is condensed by cell histones inside virion. We have talked about histones before, the concept of core histones, these basic proteins that DNA wraps around it to form a nucleosome. We'll talk about it more in the future. Polyomoviridae family of viruses have an icosahedral capsid. They have 3 structural proteins, and the size is 45 nanometer. Replication of these viruses occurs in nucleus, and once the viral replication initiates, that also stimulates host cell DNA synthesis as well. Moving to slide number 17. The genus is polyoma virus. Polyoma, many tumors, many. So these viruses cause tumors in experimental animal models. So the first virus in this family is called SV40, or simian vacuolating virus, that was isolated from monkeys in 1960s. It's the common primary cause of respiratory tract infection. 70 to 80% of adults, by the time individuals reach their adulthood, are seropositive, meaning that they have had a viral infection, and their immune system, the B lymphocytes of the immune system, have recognized viral antigenic determinants. B cells have undergone clonal expansion and differentiation and maturation to plasma cells, and they have secreted antibodies, long-lasting antibodies that remain in the serum. So if we take the serum and test it, it would be positive for SV40. SV40 is an example of a virus that causes latent infection and may become reactivated. Another virus in this family is JC virus. It was isolated in 1970s from patients with neurologic disease. Another virus in this family is called BK virus, that was isolated in 1971 from those patients who had undergone kidney transplants. Moving on to slide number 18, SV40 infections. In primary monkey kidney cell culture, SV40 causes acute as well as persistent infection. Whereas in many experimental animal models, where these animals or hosts lack some enzymes or other requirements for viral DNA replication, the disease that we see is called non-permissive infection or abortive infection. Abortive infection, as I have defined it at the bottom of this slide, is a type of viral infection that infects a cell. Virus infects a cell, but it cannot reproduce and make more infectious viral particles. The third type of infection that we see in newborn hamsters as a host, we do not see a lot of viral replication. However, viral DNA will transform the cells, or it causes tumors. Moving to slide number 19. So SV40 classification of type of infection based on the animal host. If it's in monkey, we see either acute and productive infection the virus is infectious virus. In monkeys, it can also cause persistent or long-term chronic infection or persistent latent infection that the virus can become reactivated. And in hamsters, we see a non-productive infection, that there is no viral DNA replication going on, but it can result in transformation or carcinogenesis or cancer formation of the host cells. Moving to slide number 20. I guess I will stop right here on slide number 20. We will resume our lecture on SV4D genome for next session. Thank you so much for your attention. As always, please feel free to either email me or post a comment on Canvas if you have any questions about the material we covered. Have a wonderful day.